Welcome to a revision screencast on carbohydrates. Okay, taking you right back to GCSE, what you must quickly remember about carbohydrates. Carbohydrates would have consisted of the elements carbon, hydrogen, oxygen. Okay, also just building upon that, they have a general formula of CN H2N ON, okay, where N is the number of carbons. Okay, so in biology we come across three types of basic carbohydrates. Okay, we come across trioses. Okay, so the formula for a trio would be C3H6O3, or pentoses. Okay, we come across the pentose when we study DNA. Ribose and deoxyribose are examples of pentoses. C5H10O5, and then primarily the type of carbohydrate we're going to study here, which are the type of carbohydrates we tend to find in our diet, are called hexoses. Okay, so hexose sugars would have a general formula of C6H12O6. Okay, and other words to throw at you, which you remember from when we studied this, is that these hexose sugars, these 6-carbon sugars, C6H12O6, okay, there are a number of different structural isomers of these hexoses. Okay, so words of these sugars, alpha glucose is one, or glucose you've heard of. We'll talk about alpha glucose. Beta glucose is another one we'll talk about. But there are other ones that you come across, and you've heard about fructose, and there's also one called galactose. All of these are examples of hexose sugars, and they've all got the formula C6H12O6, and they're all isomers of each other, i.e. they've got the same combinations of carbon, hydrogen, oxygen, but they're put together in different combinations. Okay, structural isomers. Okay, so these structural isomers form simple sugars, and simple sugars bind together to form complex carbohydrates, which are essentially polymers, which are chains of these sugars. Okay, so we the hexose can form what we call monomers. Okay, and a monomer in this case is given the term monosaccharide. Okay, so monosaccharides, two monosaccharides together can form a thing called a disaccharide. Okay, and many monosaccharides in a chain form what we call a polysaccharide. Okay, and the polysaccharides that you have to know about are starch, glycogen, and cellulose. Okay, so what we'll do is we'll build up the picture from very basic beginnings of what an alpha glucose molecule looks like. Okay, and then we'll show you some variants on that. Okay, so if I scroll down, here is a molecule of alpha glucose. Okay, and essentially these are carbon atoms, oxygen atoms, hydrogen atoms, and this is how it's all put together. Okay, and that's what it looks like. Obviously that's quite a complicated thing to remember, so we have a simplified version of that we can draw. Okay, and this is something that you need to know, need to remember. Okay, so what I do is I draw a basic structure like that. Okay, and what this shows you, well, that's not very neat, is it shows you all these bits here are where the carbon atoms would be. So there's one carbon atom there, second carbon atom there, third carbon atom there, fourth carbon atom there, fifth carbon atom there, and sixth carbon atom there. Okay, and in terms of how that relates to this structure up here, So in this structure here, we've got the first carbon atom here, second, third, fourth, fifth, and sixth up there. Okay, so the simplified version of that, which again you have to learn how to draw, is that. Okay, so that's called alpha glucose. Okay, and we'll go on to look at beta glucose in a second. Okay, the key thing is for alpha glucose, the arrangement of the hydrogen and the OH group, okay, around carbon 1 is that the hydrogen sits above that carbon 1 and the OH group there sits below. And the way I remember that is that this is the structure of alpha glucose. Alpha glucose. Okay, and for alpha glucose, the hydrogen on carbon 1 is above. Alpha begins with A, and obviously the word above begins with A, so alpha for above. Okay, so that's the structure of alpha glucose. Okay, so just going back to the slide, okay, so there's not an awful lot to know on this slide, is that you just need to know that alpha glucose is a hexose. Okay, so I've explained what a hexose is, a 6-carbon sugar. Okay, and these can form polymers called polysaccharides, and a monomer, okay, for a polysaccharide, we call that a monosaccharide. And you should also be aware, and I'll just write it in here, of the word disaccharide, okay, which is simply two sugars. Okay, so what we're going to do is now is to move on to the next slide. Okay, the next slide, this says to state the structural differences between alpha and beta glucose, and here we've drawn alpha glucose again, okay, in the way that exactly the same way as I drew it on the slide before. Okay, so it's first carbon, second carbon, third, fourth, fifth, and sixth. Remember, the hydrogen atom is above that first carbon for alpha. Okay, and there is an isomer of this which is called beta glucose. Okay, and here's a beta glucose molecule here, and the big difference, the only difference here, if you spot the difference, is that the hydrogen and the OH group here are the other way around. Okay, and the way I remember this is the hydrogen for beta is below. Okay, so beta for below, alpha for above. Okay, that's the way I remember it. But everything else, okay, the arrangement of the hydrogen and the OH around carbon 4 is exactly the same, okay, as it is here and here. It's only the arrangement of the hydrogen and the OH group on the carbon 1 that differs. Okay, so alpha and beta glucose are isomers of each other. For alpha glucose, the H on carbon 1 is above. For beta glucose, the H on carbon 1 is below. Okay, so that's very simply what we need to know for that slide. Moving on. So this slide looks at how monosaccharides react with each other to form bonds which holds monosaccharides together to form either a disaccharide, which is 2 monosaccharides bound together, or polysaccharides, which are many monosaccharides held together. Okay, and essentially what happens is formation of here we have 2 alpha glucoses here, okay, and what happens is a thing called a condensation reaction occurs. A condensation reaction occurs which removes a molecule of water from between these 2 alpha glucose molecules. Okay, so a molecule of water is produced in a condensation reaction. Remember, if you're having a shower, water suddenly appears on the window, okay, and we call that condensation. And again, we put these 2 glucose molecules together and water suddenly appears, condensation. Okay, so we end up, that happens, and a bond forms, okay, a bond forms between our 2 alpha glucose molecules there, okay, and that bond is called a glycosidic bond. okay, a glycosidic bond. Okay, so that's a key word. Obviously, as you remember, there's other bonds we learn for proteins and fats and DNA and that sort of thing, but the bond associated with monosaccharides binding to each other is called a glycosidic bond. And what's more, we can call this bond a 1,4 glycosidic bond because it binds between the carbon 1 of this alpha glucose molecule and the carbon 4 of this alpha glucose molecule. Okay, so this is a 1,4 glycosidic bond. Okay, so a reaction occurs between the OH hydroxyl group of carbon 1 of 1 alpha glucose, which is this one here, and the OH hydroxyl group of carbon 4, okay, that's the carbon 4 of another glucose, and a water molecule is produced. Okay, the reaction is called a condensation reaction. Remember, water suddenly appears like it does in the shower, condensation on the window. Okay, and the result is we produce a molecule which has got two monosaccharides together, and such a molecule is called a disaccharide. Di for two, okay, disaccharide. And in this case, because the two monomers, okay, are alpha glucose, this particular disaccharide is called maltose. Okay, there are other disaccharides that we come across. Okay, the obvious one is sucrose, which is the one that you put in your tea. Okay, and that's a disaccharide which contains a molecule of alpha glucose with a molecule of fructose. Okay, so it's just two monosaccharides, and that makes sucrose. Okay, and the other one you get is something called lactose, which is a molecule you find in milk, and lactose consists of one glucose molecule and one molecule of another monosaccharide called galactose. Okay, so those are three disaccharides you should perhaps have heard of. Maltose, you should really know what the structure of maltose is, and sucrose, which is alpha glucose and fructose, and lactose, which is alpha glucose and galactose. Okay, so those are disaccharides, but these molecules can also form long chains because we can add more glucose molecules here and here to form a great big long chain. Okay, and when we've got a big long chain of many monosaccharides, we call that a polysaccharide. Okay, and alpha glucose forms a polysaccharide called amylose, which forms part of starch. Okay, and I'll explain what the structure of starch is in a minute. Okay, so I've said that amylose, a component of starch. Okay, the other thing one should be aware of here is that we can break these bonds, okay, by adding a water molecule to this bond, which breaks that bond. Add water, breaks that bond, and will release our two monomers, our two monosaccharides, and that reaction is called a hydrolysis. Hydro meaning water, lysis meaning split. Okay, so we split the bond here by adding a water molecule. Okay, and that breaks the bond. How do we split that? Okay, we can either boil this up in acid, is one way we can split that bond. Alternatively, enzymes. Okay, so an enzyme called maltase, okay, which would split this bond. Okay, and starch is broken down by breaking these glycosidic bonds by the enzyme called amylase, which you've come across before. Again, that catalyzes a reaction where we add a water molecule to this glycosidic bond, splitting it, okay, to form our two separate molecules there. Okay, and that's called a hydrolysis. Okay, so the two reactions you should know are hydrolysis and condensation, and they're the opposite of each other. Condensation is when we remove a water molecule to form the bond. Okay, and hydrolysis is when we add a water molecule to split the bond. Okay, I've got a little animation which shows a formation of a glycosidic bond and also the hydrolysis of a glycosidic bond. Okay, so here we have two alpha glucose molecules. Okay, they are Perhaps it's a bit of a simplistic diagram. They've missed a little oxygen that should be here and here. I'll just draw those in. And so carrying on with that animation. So our two molecules are moving towards each other. Okay, what happens is a molecule of water is produced, okay, and the water molecule comes away, and then this oxygen bridge, as it's sometimes called, okay, holds these two monomers, these two alpha glucose molecules together, okay. Okay, and the opposite of that is called a hydrolysis, okay, which is when we add a water molecule which splits the glycosidic bond, okay, and we get back to where we started here, okay, so we've added our water molecule to here, okay, so we've got our two hydroxyl groups here, okay, by adding two hydrogens and an oxygen to that glycosidic bond that was there, okay, so that's condensation reaction to make a glycosidic bond, okay, and then splitting that glycosidic bond by adding a water molecule, and that's called hydrolysis. Okay, all those words are important to remember, and it's important to be able to draw the formation of a glycosidic bond. Okay, moving on to the next slide. Okay, this slide looks at compare and contrast two different polysaccharides that we find in plants. First one is amylose. Okay, there's actually two types of polymer that we find in starch, amylose, and another one called amylopectin. The syllabus doesn't seem to want you to know much about amylopectin, so we'll concentrate on amylose, okay, but you can find out a little bit about amylopectin in your textbook, okay, and we'll also look at cellulose. So first of all, if I look at amylose, okay, so that's a component of starch. It's a polymer of alpha glucose, okay, and it's formed solely by 1,4-glycosidic bonds, and that creates a sort of helical structure. And the advantage of that, it's very compact, so it doesn't take up much space in your cell, okay, not much volume used up in your cell because it's very compact, okay, because it's a storage molecule. It's nice that it doesn't take up much room, and also the fact that it's compact, it's actually insoluble, okay, and if it's insoluble, that doesn't affect the water potential of your cell, and that's important too, okay, and that's what it does, is it acts as a store of glucose molecules, and those glucose molecules can be released, okay, for use in respiration in your plant cell, okay. Whereas cellulose, on the other hand, is a polymer of beta-glucose, okay, and I'll show you that when beta-glucose molecules form 1,4-glycosidic bonds, each alternate glucose residue is inverted, and I'll explain why that is in a minute, okay, with the diagram below. But the result of that is it forms straight chains as opposed to these helical strains of amylose, and those straight chains bind to neighbouring straight chains by hydrogen bonds, and those form little microfibrils, and then microfibrils then bind to each other to form things called macrofibrils, and these macrofibrils bind to form cellulose fibres, okay, and cellulose fibres make a sort of meshwork which creates the structure of cell walls, okay, and those cell walls are hugely strong, okay, and but they're also, there's lots of space between the fibres, so they're fully permeable, okay, so they don't prevent molecules from getting in and out of plant cells, okay, and also the gaps between the fibres allows movement of water, the apoplast, which you probably come across in F212. Okay, so I'll explain all that in a little bit more detail as I go through some diagrams, okay. So first of all, dealing with amylose, okay, amylose is a polymer of alpha glucose. So here are lots and lots of alpha glucose molecules, well, not lots, 1, 2, 3, 4, 5 to be exact, 5 alpha glucose molecules all bound together with alpha 1,4 glycosidic bonds. Okay, so here's one carbon 4, carbon 1, carbon 4, carbon 1, carbon 4. Okay, and they're glycosidic bonds. Okay, and because of the bond angles, for those of you who are chemists involved, what that means is that this structure forms a helix, a helical structure, a coil type structure. Okay, so if you can imagine these representing each of the glucose molecules, what happens is that forms a helical structure here, and hydrogen bonds hold all of these coils, if you like, together, okay, and make the whole structure insoluble. Okay, so that's a helical structure of amylose. Okay, the advantage of this helical structure is the compactness, makes it very compact. Okay, and because the hydrogen bonds form with each other, it means that it's insoluble. Okay, doesn't alter the water potential of the cell. Compact, doesn't take up much space in the cell. Okay, so that deals with amylose. Moving on to talk about cellulose, is one of the things I said that's 1,4 glycosidic bonds with cellulose creates a bit of a problem. Okay, because of the hydrogens and the oxygen, the OH groups being on different planes, what we see is that for these bonds to form, each alternative beta glucose residues is inverted. Okay, so if you notice, this one here is with the carbon 6 sticking up, whereas this one it's sticking down. This one it's sticking up, this one's sticking down, up, down, up. Okay, so each alternative beta glucose residue is inverted. Okay, the advantage of that, the advantages with glucose, the structural significance of that, is that these form straight chains rather than the helical structure we saw up here with amylose. Okay, and what happens with those straight chains, we've got straight chains here, is that hydrogen bonds form between neighbouring chains. Okay, so these are hydrogen bonds here forming between neighbouring chains. Okay, and that creates a very strong structure. Okay, so how that works in terms of the building that up, okay, so there's a rather nice diagram here, builds the whole story up. Okay, so here I have these straight chains of these 1,4 beta glucose, 1,4 glycosidic bonds between beta glucose residues, and here are the hydrogen bonds holding these straight chains together. Okay, if we work that back, these strains form these strands of cellulose molecules which are all bound together, and they form these little microfibrils. Okay, and actually these microfibrils then bind together to form actual macrofibrils, which are things which are large enough for us to see with an electron microscope. Okay, so that meshwork of these cellulose fibres, okay, which are made up of lots of macrofibrils, which in turn are made up of microfibrils, which in turn are made up of strands of cellulose held together by hydrogen bonds, create these structures in cell walls. Okay, so that's how it works. Okay, and so going back, hopefully that's illustrated all the differences. Okay, going back to the apoplast actually, just before I move on. So talking about the apoplast, because of the gaps that occur between these cellulose fibres, okay, water can seep between these gaps, okay, which forms the main route through which water passes through plant cells, okay, by soaking round the cellulose of the cell walls. Okay, water passes through the gaps. Okay, also the gaps allow the cell wall to be fully permeable, so molecules can pass through the cell wall. Okay, so moving on to the next slide. This looks at another polymer called glycogen, okay, sometimes known as animal starch, because it's the storage carbohydrate, the storage polymer or polysaccharide we have in animal cells. Okay, so our liver cells, muscle cells, etc., have lots of glycogen stored. Okay, and the main difference between this and amylose is, as well as having the 1,4 glycosidic bonds, so this bit here is exactly like amylose, but every now and again we get this structure here. We get a branch, okay, and this is caused by a 1,6 glycosidic bond, and that creates a branch. Okay, so we get this branching effect. Okay, and the advantage of this, okay, is that we get a compact structure, much in the same way as amylose. Okay, but also the key thing here is that we end up with lots and lots of end glucose residues. Okay, so every time there's an end of a chain, there's a glucose molecule in the end of that chain, and that can be released by enzymes by the cell and used in respiration. Okay, so this glycogen molecule can give up a lot more glucose very quickly than an amylose molecule that's only got one glucose molecule at each end of its long chain, whereas it's got many end glucose terminals. Okay, so I get a nice diagram which illustrates all that down here. So here's our normal glucose. Okay, so this is it bond together, much the same as amylose, and here's one of our 1,6 glycosidic bonds forming a chain, and then lots of 1,4 glucose chain forming off that. So we get these helical structures, okay, of amylose type structures, but then branches of more helical structures coming off that. And the effect of that creates a structure a bit like this. Okay, so you can see all these lots and lots of branch chains. I get this great, rather pretty structure here. Okay, and all these end terminals around the outside of these, where there's a glucose molecule at the end of each of these chains. Now you can imagine enzymes can chop those glucose molecules off and release lots of glucose at any one time for respiration. Okay, so because animal cells are more active, so a muscle cell uses a huge amount of glucose when it's contracting to provide energy from respiration. Okay, so it needs to get access to glucose molecules of fuel very quickly, as opposed to plants. Okay, they don't respire as much as animal cells, and therefore they don't need as much glucose or be able to release glucose as quickly as we do in a plant cell. Okay, so that explains the structure of glycogen. Now, moving forward to talk about the next slide looks at explain how the structures of glucose, starch, glycogen and cellulose relate to their function in living organisms. Okay, I've alluded to a lot of this as we've gone through the last slides, but I'll repeat some of these again. Okay, so dealing with glucose. Okay, if I quickly go back to the structure of glucose, here's a glucose molecule. Okay, remember that the version of this that you have to remember is a lot simpler than this. Okay, but the glucose molecule, why is it structured as it is? Okay, first of all, it's got lots of carbon-carbon bonds. Okay, and also carbon-hydrogen bonds. Okay, and the great advantage of that is that when we break these bonds, okay, to form water, okay, we oxidize the hydrogens to form H2O water and the carbons to form carbon dioxide. Okay, what happens is that is a very exothermic reaction releasing lots of energy. Okay, so it's a fantastic substrate for respiration because energetically, when you break all these bonds to form water and carbon dioxide, we release lots of energy. Okay, so that's its principal role as a respiratory substrate. Okay, something we use for respiration. Okay, the other great advantage of this as a monosaccharide, all these OH is here, okay, make the glucose molecule soluble in water. Okay, and that's important because it's glucose in blood, for example, is the mechanism, is the way that we transport carbohydrates around our bloodstream in a soluble form. Okay, glucose, and the reason it's soluble is because of all these OHs. Okay, and those OHs can form interaction with the water molecules. Okay, therefore it's soluble in water and it can be transported in blood plasma. Okay, so those are the relating structure to function for glucose. Okay, moving on to starch. Okay, so I had this picture here of a starch molecule. I think I explained this before. The helical structure makes it compact. Okay, so it doesn't take up much space or much volume in your cell. Okay, a storage molecule, and also because the OH bonds form hydrogen bonds between sort of intramolecular bonds. Okay, water molecule can't get in there, so it forms an insoluble molecule. Okay, and that has no effect on the water potential of the cell, which is important too. Okay, so that's structure to function for amylose. Okay, and moving on, glycogen. I actually haven't put a picture of glycogen, but I had a nice picture on the slide before of glycogen. That branch structure is nice and compact, so it doesn't take up much room in the cell. Insoluble, so it doesn't affect the water potential of the cell, which is hugely important for animal cells. And also the branch structure creates lots of end glucose residues, which can be easily released for respiration. Okay, and animal cells respire more than plant cells. Okay, so they need more glucose molecules to be released more easily, and they achieve that by having these branched molecules. Okay, so that was described on the slide before. And then lastly here, the cellulose. Here's another nice diagram I found for cellulose. Here's our straight chains of cellulose forming strands, and those strands are all bound together by hydrogen bonds to form microfibrils. Microfibrils are then arranged into other structures called macrofibrils. Okay, so here's a microfibril and here's a macrofibril, and then the macrofibrils are actually arranged into these structures here, which we can actually see with an electron microscope. Okay, which is a cellulose fibre, and here's an electron microscope which shows all the gaps, this meshwork of these fibres and all the gaps that occur between them. These allow water to permeate through this cellulose cell wall, and also water to soak through it, the apoplast route in transport in plants. Okay, so just going through some of that below on your slide. So cellulose and plant balls, straight chains of beta glucose molecules form microfibrils and macrofibrils, and those are bound strongly by hydrogen bonds between neighbouring straight chains of these beta glucose polymers. Okay, the cellulose has great strength. Okay, all those hydrogen bonds hold it together, forms a great strong structure, and that's important for turgor pressure. Okay, when you did that with in F211, so the cell membrane full of water pushing out against the cell wall, that creates turgor pressure, which is very important in support in plants. And the cellulose macrofibrils enable cell walls to be fully permeable. Okay, so the gaps between the macrofibrils allow water and any other solid to pass through, and therefore it's fully permeable, as opposed to the semi-permeable nature of the cell membrane, which is the phospholipid bilayer. And the cellulose macrofibrils will allow water molecules to soak along them, okay, which is the apoplastic route in root and leaf, which you have studied in F211. Okay. So that finishes our look at carbohydrates. There's also tests on carbohydrates, the Benedict's test and the iodine test, and I'll come back to that point on a subsequent screencast which looks at tests for biomolecules.